The second part is about environmental factors. There are numerous environmental factors can contribute to the development of pickles. Let's take a few common examples from life, like toxic chemicals, drugs, microbiomes, and exercise. BPA is a chemical produced in large quantity for use primarily in the production of polycarbonate plastic. It is found in various products, including shutter-proof windows, air wires, water bottles, and epsom resins that coat some metal food can, bottle top, and water supply pipes. It is one of the highest volume chemicals currently available, known to act as endocrine disruptor and alter hormone profile and several metabolic functions, including inflammatory pathway. Elevated levels of BPA have been found in women with PCOS. In experimental animals, neonatal exposure to BPA resulting in a polycystic ovary-like syndrome in adulthood. Recently, we found that BPA is involved in regulating cholesterol metabolism, mainly through ABCA1 gene, to affect reproductive endocrine function. ABCA1 is an ATP-dependent membrane protein that promotes intracellular cholesterol and phospholipid efflux. Another classical environmental factor is VPA, which is a broad-spectrum anti-leptic drug. Common adverse effects of VPA are PCOS-like change, hyperandrogenism, menstrual cycle change, PCOM. We recently found that VPA commonly affects many steroid metabolism-related genes. It was interesting to note that the vitamin D receptor gene was significantly upregulated. Several studies indicated that women with PCOS have the lower level of vitamin D. Maybe vitamin D supplementation is an effective treatment modality for reducing the effect of VPA on PCOS. When I studied for my PhD, my first journal club topic is this medical hypothesis, dogma theory, a novel theory for the development of PCOS. Disturbance in bacterial flora brought about by a poor diet created an increase in gut mucosal permeability, with a resultant increase in the passage of LPS gram-negative colonic bacteria into the systematic circulation. The resulting activation of the immune system interfered with insulin receptor function, driving up serum insulin level, which in turn increased the ovary production of androgen and interfere with the normal follicle development. 7 years later, in Nature Medicine, Chinese scientists confirmed the theory and suggested that modifying the gut microbiota, altering bile acid metabolism, and interleukin-22 level may be of value for the treatment of PCOS. The father of Western medicine, Hippocrates, also said that all diseases begin in the gut. It makes sense. The last environmental factor I want to introduce is sport. 27% of athletes not taking birth control pills experience irregular menstruation. The most common diagnosis associated with menstrual dysfunction was PCOS, and not hypothalamic inhibition because of energy deficiency. There are 3 kinds of sport type: power, endurance, technical. Interestingly, PCOS was listed in the power group. You can see the classification of the Olympic sport event from this table. The Lancet published a study looking at the relationship between different types of physical activity and all-cause mortality through a 10 years follow-up of 80,000 people. The 3 exercises turned out to be the most cost-effective. So let's go to play badminton. After discussion of environmental factors, let's move to the genetic factors. PCOS has a familial aggregation with a heritability of about 72%, and genetics is the main causal factors. The genetics of PCOS has progressed through family studies to candidate gene studies to genome-wide association studies, GWAS has greatly advanced the genetics of the disease. 10 years ago, our team, led by Professor Yang Chen, has carried out the first 2 PCOS GWAS in the world. We identified 11 PCOS susceptibility regions within the genome, including the traditional gene like F8 receptor and LHCGR, the new gene like the THADA-DIN C9ORF3. PCOS susceptibility loci are mainly involved in the oral response to the gonadotropic hormones, the metabolic of glucose and lipid, and cell cycle regulation. Our study was also described as the major milestone in the history of PCOS genetic research. Our findings have been widely validated in European and American populations, which suggests that PCOS has the same genetic background. In other words, PCOS is an Asian disease. In 2018, a large-scale genome-wide meta-analysis of PCOS suggests shared genetic architecture for different diagnosis criteria like self-report PCOS, PCOS diagnosis by NIH, or Rotterdam criteria. Genotype and phenotype analysis found that each symptoms require multiple genetic variants to explain, and each genetic variant explains only part of the syndrome. It suggests that PCOS is a polygenetic disease. GWAS is an observational study. In recent years, Mendelian randomization study was used mainly for the epidemiologic etiology interference. Mendelian randomization studies suggest a variant associated with BMI, fasting insulin, menopausal timing, depression, and male pattern volume play a causal role in PCOS. This is the overview of Mendelian randomization study involving PCOS. PCOS as the outcome or as the exposure are summarized with significant results at the top of the figures. Please learn by yourself. Next, I will talk about PCOS evolutionary paradox. PCOS is an ancient disease with a high genetic predisposition, and it's markedly impaired reproductive function, raising the paradox of why the disease has not disappeared rapidly over the generations, but has maintained a high incidence worldwide. Let's have a look from the genetics. One study has analyzed the PCOS phenotype-genotype relationship in silicon using SNPs or representing genes for analysis of genetic clustering and distance to evaluate the degree of genetic similarity. This figure is about the world distribution of the affinity to the genetics cluster and PCOS phenotype's prevalence. PCOS individuals were assigned to 5 genetic clusters, matched with different world regions. You can see SNP-based Bayesian genetic clustering reveal closer association between PCOS genotypes and the metabolic phenotypes in Chinese population. As we know, contemporary human genetic study informed us that the origin of human ancestor is from Africa. Ancient humans came out of Africa 3 times but were unable to adapt due to the climate and all became extinct there, and only the 3rd time they succeed. From an evolutionary pathogenic mechanism underlying PCOS might be candidate factors for survival advantage of the human beings. Former compensatory advantage factors may become pathogenic mechanism underlying complex metabolic disease with prolonged life expectancy and transition to sedentary lifestyles. Previously, our transmission disequilibrium test found that 2 of these GWAS loci were over-transmitted in co-families. Its related gene is THADA and C9ORF3. From the evolutionary perspective, we can see these 2 gene-related SNPs have protective effect on PCOS, and the literature and our preliminary data also support that the corresponding genes are closely associated with energy metabolism. Previous study identified that the Theta gene as the major effect locus by positive selection in the evolution of modern humans adapted to cold. So, let's take Theta as an example to expand. SNPs in the Theta also be associated with type 2 diabetes and BMI. The encoded protein, thyroid adenoma-associated protein, is likely involved in the death receptor pathway and apoptosis. It is a kind of a putative death receptor fusion protein. Many DOFs are highly conserved, indicating an important role in biology. However, many such DOFs are not essential genes. Their biological roles often remain unknown. For instance, DOF 143 is present in most bacteria and eukaryotic genome. However, when it was deleted in E. coli, no obvious phenotype was detected. Later, it was shown that the protein that contain DOF 143 are ribosome splicing factors that blocking the assembly of the two ribosome subunits. While this function is not essential, it helps the cell to adapt to low nutritional conditions by shutting down protein biosynthesis. As a result, this protein and the DOF only became relevant when the cell starved. It is thus believed that many DOF are only required under certain conditions. While this function is not essential, it helps the cell to adapt to low nutrition conditions by shutting down protein biosynthesis. As a result, this protein and the DOF only became relevant when the cell starved. It is thus believed that many DOF are only required under certain conditions. In adipose, depletion of Theta gene causes significant retardation of growth, hypersensitivity towards low temperature, and a lower energy charge. In Drosophila, Theta knockout leads to obesity, accompanied by hyperphagia, reduced thermogenesis, and an increase in lipid storage, mostly in the fat body. Interestingly, the female phenotype was more pronounced. As the evolution of Theta is associated with adaptation to colder climate, these findings are especially interesting in the context of human evolution because they uncover a link between the evolution of Theta and the associated adaptation in energy metabolism. As Theta could regulate the organism balance and between energy storage and heat production in plant and fly, further characterization of the metabolic effect of Theta in mammal and a better understanding of its regulation might provide a new therapeutic target for the treatment of reproductive and metabolic disease like PCOS. We are working on it. We have obtained a large number of very interesting results in mice and humans, and look forward to publishing them soon and share them with you. These findings also support the idea that genetic triggers leading to PCOS might be candidate factors for the survival advantage of the human being. Although natural selection can eliminate deleterious genes resulting in the harmful predisposition of the living organisms, the persistence of susceptibility gene as a risk factor for metabolic disease may be due to the fact that the rapid change in human lifestyles may not have allowed enough time for any compensatory adaptation. To gain insight into PCOS, we need to rethink in evolutionary perspective, take genetics and environmental factors into account, especially the impact of rapid change in lifestyle. Welcome to join us to explore the puzzle of PCOS. Thank you for your listening.